Hello everyone, welcome back to my channel Bunny's Biology. Today's lecture on mutation. So mutation are changes in our DNA that can save evolution, cause disease or sometimes have no effect at all. So in this lecture we will explore what is mutation, different type of mutation and their real world impact. So what is mutation? So a mutation is an permanent alteration in genetic material, that is DNA or RNA that can be passed to the future cell or offspring. So this is the normal gene and this is the mutated gene. So in the mutated gene where the nucleotide is changes or a change of part of gene or change in chromosomal structure or number that altered the abnormal protein or no protein. So mutation may occur spontaneous during DNA replication, this is the DNA replication, or induced by some external factor such as radiation, chemicals or infectious agent. In a multicellular organism, a mutation may occur in any cell and at any stage during development. So mutation occur in both somatic and germline cell. So what is somatic cell and what is germline cell? Somatic cell, that is body cell that cannot participate in fertilization, and germline cell is that is reproductive cell, that is sperm or ovum that participate in fertilization. So when the mutation occur in the body cell, that is called somatic mutation, and when the mutation occur in the germline cell, that is germline mutation. So in somatic mutation, DNA change occur in body cell, not sperm or egg after fertilization, and that cannot be passed to children, that is non-hereditary, and affect only the mutated cell and its descendant. And this is the parent gamete, embryo, organism. Here patch of the affected area, this is the affected area, and this is the body cell, and that cannot be passed to children. Germline mutation is DNA change that occur in reproductive cell, that is sperm or ovum. Because this cell participate in fertilization, the mutation become part of entire genome of offspring. And in germline mutation, that is passed from parent to child and present in every cell of the offspring body. And germline mutation in sperm, that's why entire organism carry the mutation. So let's explore the different type of mutation. So one is gene mutation and another is chromosomal mutation. So what is gene mutation? So gene mutation is the change in nucleotide sequence of a single gene. So this is the chromosome, and the chromosome are composed of long, tightly coiled DNA molecule. So gene mutation is the change in the nucleotide sequence. DNA is consist of 4 nucleotide: adenine, guanine, cytosine and thymine, and this change in the nucleotide sequence of single gene is called gene mutation. But chromosomal mutation is the change in the structure or number of entire chromosome. This is the chromosome, and when the change in structure, this is the changes of structure or entire chromosome number, that is called chromosomal mutation. So this chromosomal mutation can involve large segment. This is the large segment of DNA or even whole chromosome. They often have major effect because they can disrupt many genes at once. So let's understand the various types of gene mutation. So first is the point mutation, that also called base substitution mutation, where the one nucleotide is replaced by another nucleotide. This can result in 3 outcomes. One is silent mutation, another is missense, other is nonsense mutation. So first is silent mutation, where the codon is changes but amino acid remain the same. So we all know that this is the chromosome, and the chromosome are composed of long, tightly coiled DNA molecule. This is the DNA molecule, and the DNA synthesized of mRNA by the process of transcription, and the mRNA synthesized protein by the translation process. So a protein is a large biomolecule consist of long chain of amino acid joined together. So long chain of amino acid is called polypeptide, and that form the large biomolecule, that is called protein. So one amino acid specify each codon. So what is codon? Each codon is a sequence of 3 nucleotide. This is 3 nucleotide, and each codon is a sequence of 3 nucleotide in mRNA. Like GAG that code glutamate. So one codon that specify one nucleotide. But when a mutation alter the DNA sequence, so there is the mutation happened where the change the one nucleotide, and that create a new codon, and that may code for a different amino acid. So in silent mutation, where the codon changes but amino acid remain the same, this mutation is called silent mutation because of silently mutated, no changes in the cell because of the amino acid is same. So here is the example is CAA that codes the glutamine. After change the CAG, A changes into G, that also the same amino acid codes, that is glutamine. So this mutation is called silent mutation. Next is the missense mutation, where the codon changes and also changes the amino acid. Like example, this is the normal and this is the missense mutation, where the GAG that code the glutamate. But after changes the GAG to GUG, changes happened in the DNA, where the GAG A changes into T. Then after mRNA, this GAG codes the glutamate amino acid and GUG that code the valine, and that effect in the hemoglobin molecule, that effect in the structure of red blood cell. Red blood cell is changes. And this disease is called sickle cell anemia, where the glutamate changes into valine. And this mutation is called missense mutation. Next is the nonsense mutation. In this mutation, stop codon is appeared too early and truncating the protein, where codon is changes and this change into stop codon. So what is stop codon? Stop codon mainly UAA, UAG and UGA. This 3 codon is called stop codon that signal the end of translation process. So this stop codon mainly happened in the nonsense mutation, where the codon changes into stop codon. So here is the example where this is the normal sequence where the normal protein is produced. After the nonsense mutation, where the CGA changes into UGA, where C change into U, and this CGA that codes the arginine amino acid. After nonsense mutation, this CGA transform into UGA that code the stop codon. Here the protein length is shortened, that is called truncated protein. So here is the protein length, and after nonsense mutation, the protein length is shortened. So for the nonsense mutation can cause different type of disease like cystic fibrosis. In this disease, premature termination happened by the premature stop codon. This is the premature stop codon, and that results shortening the protein. This is the shortening of protein, and that usually cannot function properly and leading to disease. So another 2 specific type of point mutation is transition and transversion. So in the transition mutation occurs when a purine is replaced by another purine, like purine is adenine, guanine, and pyrimidine is cytosine and thymine. So transition is purine replaced by another purine. So A to G or G to A. So pyrimidine is replaced by another pyrimidine, like C replaced by T or T replaced by C. So this process is called transition mutation. So transition are more common because the chemical structure is similar. So this is the purine, this is the purine. So chemical structure is similar. But in transversion are less frequent but more disruptive. So transversion mutation occur when a purine is replaced by pyrimidine or pyrimidine is replaced by purine. So here adenine guanine, that is purine, and cytosine thymine, that is pyrimidine. So adenine is purine, that is replaced by cytosine, that is pyrimidine, adenine to thymine, guanine to cytosine, and guanine to thymine. This transversion can cause significant change in genetic course because this transversion process is not more common because their chemical structure is not same. So these all the various types of point mutation. So point mutation are the simplest, but insertion and deletion can be more disruptive. So what is insertion and deletion? So an insertion are adding of one or more nucleotide in DNA sequence. So this is the addition of one or more nucleotide in DNA sequence. This can disrupt the reading frame leading to abnormal protein. And deletion mutation is the remove of one or more nucleotide in a gene. So small deletion remove one or few nucleotide within a gene. So this is the DNA where the one nucleotide is added, then it's called insertion mutation, and when the one nucleotide or more nucleotide is deleted or lost, that is called deletion mutation. Here is the example. In the insertion mutation example is the TSH disease, and the deletion mutation is the example of the cystic fibrosis. So here is the example where the insertion happened and that changes the all reading frame, that leading to abnormal protein. So this is the original DNA sequence. So in the original DNA sequence, each codon, that is 3 nucleotide, translate into amino acid histidine. So CAT continuously 5 repeat that give us protein sequence histidine. So when the single nucleotide, that is A, inserted into the DNA sequence, this shift the reading frame changing. That is CAT. After ACA, then TCA, then TCA. And this leading to completely different set of amino acid instead of histidine. Now get threonine, serine, and serine. Same as deletion mutation where the deletion of 3 base pair. 3 base pair is deleted, CTC, and that modify amino acid sequence. This is the leucine. After deletion of CTC, the amino acid is histidine, leucine. That modify the amino acid sequence. So next mutation is the frame shift mutation. So this mutation also called reading frame shift, where the mutation involve the addition or deletion of one or few base pair. And addition and deletion that occur within the coding region of gene are collectively referred to as frame shift mutation because they alter the reading frame of all bases tripled. In this picture, this is the normal DNA, and this normal DNA that produces the full length of normal protein. So after insertion of C nucleotide that disrupt the reading frame. So this is the normal CAA before the mutation. So after mutation, CAA that changes into CAC, and that codes the histidine amino acid. So after insertion of nucleotide that completely changes the all reading frames downstream of the site at which the mutation occur, and that produce a non-functional or truncated protein. This mutation is called frame shift mutation, where the changes of the all reading frame after mutation. So now that we have understood the different type of gene mutation, where mutation can alter the DNA sequence within a single gene, but when the change involve large DNA sequence or whole chromosome, we call them chromosomal mutation. Let's look at how these differ. So next is the chromosomal mutation, where mutation involve much larger structural changes, and this mutation affect the structure or number of entire chromosome because chromosome carry thousands of genes. So in the chromosomal mutation that happen numerical chromosomal mutation or structural chromosomal mutation. In the numerical chromosomal mutation, where the number of chromosome is changes. This is the normal set of chromosome where the number of chromosome is changes. Here each 2 chromosome and this changes into 3 chromosome, that is called trisomy, and this also deletion of 1 chromosome, that is called monosomy. So in structural chromosome, where the chromosome structure is changes. So let's see the different type of structural chromosomal mutation. So different type of structural chromosomal mutation. One is duplication. So in chromosomal duplication is a structural mutation where a segment of the chromosome is copied and inserted again. That like the increased gene doses or can disrupt normal development. This is the original chromosome, and in a chromosome is duplicated a segment chromosome. So here is the EF 2 segment chromosome is duplicated, that is EF. This mutation is called duplication, chromosomal mutation. So there are 3 types of duplication process. One is tandem duplication. So this type of duplication where a duplicated segment is adjacent to the original segment, like this. This is the original segment. This segment is duplicated adjacent to the original segment, and this mutation is called duplication mutation. And another mutation is the displaced duplication. So in displaced duplication, here is the duplicated chromosome is located some distance from the original segment, like this is the original chromosome. So in displaced duplication, here is the AB, this is the centromere, CD, EF, G, then EF. So here is the original segment that is duplicated into some distance from the original segment. This is called displaced duplication mutation. Next is the reverse duplication. So this is the original chromosome. So in the reverse duplication, here is the, this is the AB, centromere, CD, EF, then FEG. This is the original segment, and the duplication is reversely, then is this side. So this is called reverse duplication. So one is tandem duplication, next is displaced duplication, and another is reverse duplication. So tandem duplication, where is the duplication is adjacent to the original segment. And next is the displaced duplication, where is the duplicated chromosome is distanced from the original segment. And the reverse duplication is the where duplication of the chromosome segment is inverted like. This is the inverted like. So next is the deletion mutation. When the segment of chromosome is loose, this is the chromosome, normal chromosome where the EF segment is loose or deleted. Like example, Cry-Dusar syndrome, where this is the rare chromosomal disorder caused by deletion on the short arm. This is the short arm. This arm is called P arm, this arm is called Q arm. So in Cry-Dusar syndrome, where the short arm, that is called P arm of chromosome 5 is deleted, and that name means cry of a cat in French, refers to the distinctive high-pitched cat-like cry seen in newborn. So this is the deletion chromosomal mutation. Next is the inversion mutation, in which a chromosome segment is inverted turn 180 degree. Here is the original chromosome segment, that is A, B, C, D, E and F. This D, E, F that is inverted turn 180 degree, like D, E, F, and that may disrupt gene regulation, and this mutation is called inversion mutation. Inversion mutation is 2 types. One is paracentric paracentric and another is pericentric. So what is paracentric and pericentric? So paracentric is when the inversion do not include the centromere. This is the centromere. When the inversion do not include centromere, this is called paracentric. And pericentric when the inversion include the centromere, this is called pericentric. So here is the example A, B, C, D, E, F, G. And this is the centromere. When the inversion do not include, like 180-degree turn, like CDE turn into this like CDE. That is called paracentric inversion, not include the centromere. But in pericentric inversion, here this is the centromere. So here CDE, in between D and E present of centromere. So when CDE segment turn into 180-degree like CDE, that include the centromere. This is called pericentric inversion. So next is the translocation. So translocation is a type of chromosomal mutation where a segment of chromosome moved between non-homologous chromosome or within same chromosome. This is the one type of chromosome. This is the another type of chromosome where the segment of chromosome is moved between a non-homologous chromosome or within same chromosome. So here E, F and G is a chromosomal segment where this chromosomal segment is moved to the another homologous chromosome in the position of Q, R and S, and this Q, R, S of another non-homologous chromosome is moved to the side of E, F, G position of chromosomal segment. So this process or mutation is called translocation mutation. So translocation come in two major types. One is reciprocal translocation, another is non-reciprocal translocation. So in reciprocal translocation, where a segment are exchanged between two non-homologous chromosomes. So like this, where the one is non-homologous chromosome, another is non-homologous chromosome, where the segment of chromosome is exchanged between them. So here A, B, C, D and E. Each segment of one non-homologous chromosome is moved to the another non-homologous chromosome in the position of I. So this I segment of another non-homologous chromosomal segment is moved to the one non-homologous chromosome in the position of each segment. So here reciprocal translocation where the exchange of between two non-homologous chromosomal segment. So this is called reciprocal translocation. But in non-reciprocal translocation, where a chromosomal segment move to another chromosomal segment without any exchange. So here is the E segment of one chromosomal segment move to the another chromosomal segment. This is the another chromosomal segment without exchange. So like this, here is A, B, C, D, E. Here E chromosomal segment is moved to the F, G, H, I, another chromosome like F, G, H, I and E. So here no exchange is happened. So in reciprocal translocation, here is exchange between two non-homologous chromosomal segment. But in non-reciprocal translocation, only one chromosome donate a segment without any exchange. This is called non-reciprocal translocation. So here is the example, Philadelphia chromosome. It is the example of reciprocal translocation, where the human chromosome 9 and human chromosome 22 exchange chromosome segment. So where a segment from end, this is the long arm of chromosome 9 and this is the long arm of chromosome 22. So long arm of chromosome 9 move to the long arm of chromosome 22. Simultaneously, a piece from chromosome 22 move to the chromosome 9, creating a reciprocal translocation. And this exchange fuses the BCR gene. This is the BCR gene on chromosome 22 with the ABL gene on chromosome 9, forming the BCR ABL gene. And this chromosome is called Philadelphia chromosome. And this BCR-ABL protein fusion create a oncogenic protein. This protein signals cell to proliferate uncontrolled cell division and develop rapid production of abnormal WBC, cause leukemia. This chromosome is called Philadelphia chromosome. This is the example of reciprocal translocation. And another type of chromosomal mutation is the Robertsonian translocation mutation. This translocation is a special type of chromosomal mutation where a short arm of acrocentric chromosome exchange with the long arm of another acrocentric chromosome. So what is acrocentric chromosome? This is the acrocentric chromosome when a centromere is located very close to the end and form a very long arm that is called Q arm and another very short arm that is called P arm. This is called acrocentric chromosome. So in Robertsonian translocation mutation where the short arm of one acrocentric chromosome is exchanged with long arm of another acrocentric chromosome and the two acrocentric chromosome break near the centromere. This is the break point. And after exchanging, creating a large metacentric chromosome. So what is metacentric chromosome? This is the metacentric chromosome where a centromere is located in the middle position and two arms are almost equal in length. P arm and Q arm almost equal in length. This is called metacentric chromosome. So after exchanging short arm with the long arm of another acrocentric, creating a metacentric chromosome and a fragment that often fail to segregate and lost. And this fragment is segregate and lost. So when a person with Robertsonian translocation mutation, this person end up with 45 chromosome instead of 46 chromosome. So in human 5 acrocentric chromosome present, chromosome 13, chromosome 14, chromosome 15, chromosome 21 and chromosome 23. All are acrocentric chromosome in human. So now we have explored structural chromosomal mutation that change in shape or structure of chromosome like deletion, duplication, inversion and translocation. Now it's time to look at mutation that change chromosome number. So let's dive into numerical chromosomal number including aneuploidy and polyploidy. So aneuploidy is a condition in which the number of individual chromosome is changed. So in aneuploidy, number of chromosome is changed, meaning one or more chromosome are missing or extra. So aneuploidy is usually result from non-disjunction during meiosis 1 or meiosis 2 or chromosome fail to separate properly. But in polyploidy is a condition in which an organism has more than two complete set of chromosome, not just individual chromosome, one or more complete set of chromosome from the usual state. Usual state is generally diploid. So one or more complete set of chromosome has been gained from the usual state. This is called polyploidy. So this is the normal diploid 2n. So here diploid cell with two set of chromosome. Here chromosome 1, chromosome 2, chromosome 3 and chromosome 4. Each chromosome is present in two copy, one from each parent. So this is called diploid. So this is the polyploidy state and this is the aneuploidy state. In polyploidy where the three complete set of chromosome. So each chromosome 1, chromosome 2, chromosome 3 and chromosome 4 appear 3 times. That is called triploid, 3n, 3 set of chromosome. And tetraploid, there is 4 set of chromosome. Chromosome 1, 2, 3 and 4 each appear 4 times. So in polyploidy is a one or more complete set of chromosome has been gained. So here triploid, 3 set of chromosome. Here tetraploid, 4 set of chromosome. But in aneuploidy, where the individual chromosome are gained or lost. So in trisomy 2, here is 2n plus 1. Here trisomy 2 means chromosome 2 appear one extra chromosome added to a pair. This is the one extra chromosome that added in chromosome 2. That is called trisomy. That is 2n plus 1. But all other chromosome are normal. Like monosomy where 2n minus 1. Where monosomy 1 where the chromosome 1 where one copy is missing. That is called monosomy 2n minus 1. But other chromosome is normal. This is called aneuploidy where gain or loss of individual chromosome. So different type of aneuploidy is monosomy 2n minus 1 where is loss of one chromosome. Like example Turner syndrome. This is the Turner syndrome karyotype. Turner syndrome that affect only female. It occurs when X chromosome is completely or partially missing. So normal Turner syndrome happened in female. So normal female have 46 chromosome like 46XX. But in Turner syndrome, the karyotype is 45X or X0. One chromosome is lost. This is called monosomy 2n minus 1. And trisomy 2n plus 1 where is gain of one chromosome. Like example Down syndrome where 21 chromosome where the one extra chromosome is added in 21 chromosome. This is called trisomy. Like Patau syndrome, that is trisomy 13. In trisomy 13 where the one chromosome is added to a pair. Like Edwards syndrome, trisomy 18 where the in 18 chromosome where extra one chromosome is added. So this is called trisomy 2n plus 1. Another aneuploidy is nullisomy, that is 2n minus 2. Loss of a pair of homologous chromosome. So 2n minus 2 means loss of a pair of homologous chromosome. In diploid here chromosome 1, chromosome 2, chromosome 3 and chromosome 4. In nullisomy where fully a pair of homologous chromosome is missing means loss. That is called nullisomy. Next is the tetrasomy 2n plus 2. Gain of two homologous chromosome. So here is the tetrasomy where the two extra chromosome is added to a single pair. So this is the 4 chromosome where the two extra homologous chromosome is added. Next is the double trisomy where 2n plus 1 and plus 1. So trisomy means 2n plus 1. So double trisomy means 2n plus 1 and plus 1 where the extra copy of two different chromosome is added. Like here chromosome 3 and chromosome 4. Here one chromosome is added, here one chromosome is added. So this is the double trisomy. So extra copy of two different chromosomes. This is the chromosome 3 and this is the chromosome 4. Two different chromosome, one extra chromosome is added. That is called double trisomy. So double monosomy, 2n minus 1 minus 1, where one chromosome missing from two different set. So this is the double monosomy, where chromosome 3 and chromosome 4, where the two different pair of chromosome or two different set of chromosome from here one chromosome and one chromosome is missing. That's why double monosomy, 2n minus 1 minus 1 different. And double tetrasomy, 2n plus 2 plus 2. This is the double tetrasomy, where the two extra pair of homologous chromosome, 2 here 2. So two different chromosomal pair where two extra pair of homologous chromosome is added. So this is the third chromosome and this is the fourth chromosome. In third chromosome and fourth chromosome, two different chromosome pair where the two extra pair of homologous chromosome is added. Here is extra two homologous chromosome pair is added. This is called double tetrasomy. So these all are different type of aneuploidy. Next is the different type of polyploidy, where the complete set of chromosome is increased. So this is the haploid. Haploid cell has one complete set of chromosome, like this is the chromosome 1, this is the chromosome 2 and this is the chromosome 3, where the one complete set of chromosome, only one complete set, that is called haploid. Like example, in human sperm and ovum that have haploid number of chromosome, like n equal to 23 number of chromosome. In diploid that have two complete set of chromosome, one from each parent. So from one parent n is 23 pair chromosome, that is from sperm, and another ovum, that is n haploid number of chromosome that join with the sperm and form a 2n, that is called diploid. So two complete set of chromosome, one from each parent. This is called diploid. Next is triploid, where the three complete set of chromosome. Here chromosome 1, chromosome 2 and chromosome 3, where completely three set of chromosome. Mainly many seedless fruit like banana and watermelon that have three set of chromosome, like triploid number of chromosome is present, but rare and usually lethal in human. So like example, in human 2n means 46 chromosome. In triploid, where is the 3 set of chromosome, means 3 into n, means 69 chromosome, usually lethal in human. So tetraploid is where the 4 set of chromosome. This is present in many plants like potato, cotton, some human tissue, liver cell can be polyploid, and whole organism tetraploid in human is usually lethal. So tetraploid is 4 complete set of chromosome. Here is the 4 complete set of chromosome. So this all the different type of polyploidy. So alright, that brings us to the end of today's lesson. So we explore how mutation, whether at the gene level or the chromosomal level, shape biology in powerful ways. So mutation are the driving force behind genetic variation, evolution and many of the traits we see in living organism. So if you found this lecture helpful, don't forget to like, share and subscribe to the channel for more clear and engaging biological lesson.